Alright, so let's talk a little bit about nucleic acids. This is the fourth type of the four organic molecules we're going to discuss in this chapter. We've talked about carbohydrates, we've talked about lipids, proteins, and now nucleic acids. Remember that, and just for a quick review, the monomer of carbohydrate is monosaccharide, the dimer of carbohydrate is disaccharide, and then the polymer of carbohydrate is polysaccharide. We talked a little bit about the specific examples of monosaccharides, disaccharides, and polysaccharides. Remember that all of our polymers are linked via dehydration synthesis. We said that over and over and over. And all of our polymers are broken down via hydrolysis. Okay, these are no different. And the nucleic acids are going to be composed of a monomer. And the monomer of nucleic acid is a nucleotide. Okay, and if I highlight one full nucleotide, it is this structure right here. Okay, it's composed of three parts. The nucleotide consists of a phosphate, which is here. Okay, that's a phosphate. This would be a pentose, and pentose is a 5-carbon sugar. In DNA's case, pentose is deoxyribose. And in RNA's case, the pentose is ribose. Okay, that's how DNA gets its name. DNA is deoxyribonucleic acid. RNA is just ribonucleic acid. Okay, and so that name comes directly from the sugar that makes up its 5-carbon pentose. Okay, and then this over here would be the nitrogenous base. So really three parts: phosphate, sugar, nitrogenous base. Okay, and so this would be one nucleotide. This would be the second nucleotide. This would be the third nucleotide, and so on. Okay, notice that they all have the same sugar. They all have the same phosphate. The only thing that differs between all of the nucleotides are the nitrogenous bases. They could be either T, A, C, and G in DNA, and they could be A, U, C, and G in RNA. Okay, we're going to learn a lot more about DNA and RNA and the processes involved in DNA replication and RNA transcription later in Chapter 12, but for right now we just need to know what DNA and RNA is made of and what the structure of these molecules are. Okay, so if we think about DNA by itself, DNA, like I said, is or stands for deoxyribonucleic acid. There is the D in A of that molecule. Okay, deoxyribonucleic acid. DNA is double-stranded. You can see that here. One strand is running this way in a 5' to 3', and this strand over here is running the opposite way in a 5' to 3'. And like I said, we're going to talk about that later in the year, but for now, know that one strand runs opposite the other strand, so we get this not only double-stranded structure, but this anti-parallel construction, okay, where it's not truly parallel, because parallel lines, if you recall in math, are two rays running in the same direction forever, right? These are two rays, but they're not running in the same direction, so they're said to be anti-parallel, okay? They're still parallel, they still don't overlap, but they're running in opposite directions. Okay, so far, DNA stands for deoxyribonucleic acid. It is double-stranded. These double strands orient themselves in opposite directions, which give rise to this anti-parallel construction. DNA is only found in the nucleus. Okay, DNA is so important that it is only contained within the nucleus, and we can never remove it because it's just a scary world out there. It's wild, wild west. Okay, it's your job, or the cell's job, rather, to keep this DNA in the nucleus where it can keep it safe all the time. And you might be saying, well, then how does the cell get the instructions out to all the cellular workers and the organelles? And you're correct in thinking that way, because we do need to get the instructions out into the cell, but we send the instructions out via RNA. Okay, DNA is our kind of master plans. RNA would be copies of that master plan, kind of like recipe copies. Okay, and like I said, we're going to talk about RNA next. But for now, The sugar in DNA is deoxyribose. We've established that. DNA is only found in the nucleus. DNA, like I said over here, is only composed of adenine, guanine, cytosine, and thymine. These are the A, T, C, and G's you see over here. Those are nitrogenous bases. And like we said, DNA and nucleic acid is really good at storing genetic material. And so we say that genes are sections of DNA, and they're not the majority of DNA. In fact, they're only about 2%. Okay. A lot of what you have in your DNA is just ancestral and really not needed. Okay. Nucleic acids, RNA, are kind of the opposite. We have here DNA, and again, this is transcription. We're going to talk about this later in the year, but this shows you the DNA double helix. Sorry about that. So this shows you the DNA double helix and anti-parallel construction, right? RNA, this shows you, this is the RNA right here, okay? RNA is made from DNA, but RNA is only single-stranded, okay? It's basically, if we were to cut this DNA molecule in half long ways, half of that, meaning you would have one backbone and a bunch of floating nitrogenous bases. And you know, compare that to DNA where you have two backbones on the outside, kind of creating this twisted ladder look, and all of your nitrogenous bases form bonds on the inside of the backbone, creating this alpha helix or this double helix. Okay, so a little bit about RNA. RNA is ribonucleic acid, again RNA. RNA is composed of ribose. Ribose is the 5-carbon sugar that is in the center of this molecule. RNA, like we said here, is single-stranded. RNA is found outside the nucleus. Now, because RNA is made from DNA, RNA is made in the nucleus, but it can easily travel through one of the nuclear pores found on the nucleus and will travel out into the cytoplasm, where it will meet with a ribosome and or attach itself to well, it'll meet a ribosome, but that ribosome can be attached to either the rough ER or a cytoplasm cytoskeleton component. And we'll talk about that a little next chapter. But as long as you know that the RNA will find a ribosome in order to produce protein, then you'll be good for now. RNA is composed of 4 bases like DNA is, but there is a difference. DNA does not contain uracil, RNA does. Okay, and then there's 3 different types of RNA. You have mRNA, rRNA, and tRNA. The tRNA stands for transfer, rRNA stands for ribosomal, mRNA stands for messenger, and they have 3 distinct roles. The messenger RNA, like this molecule, takes the message from the nucleus to the cytoplasm. Okay, it is the messenger RNA. It takes message from nucleus to the ribosome. Ribosomal RNA, the rRNA, is basically the ribosome, or is a portion of the ribosome that will produce the protein by reading the mRNA message. And tRNA kind of looks like one of these little guys, and its job is to bring this circular amino acid, which we talked about last chapter, or last lecture, and pair this amino acid to a specific codon in order to link them and produce a protein. But again, we're going to talk about the biochemistry of that and the bioprocesses of that in later chapters. Okay, just know that there are 3 types of RNA at this point: messenger RNA, ribosomal RNA, and transfer RNA, and all 3 of them do a distinct specific job, and they all look different. Okay, so just another case of structure dictates function, right? This one is structured to allow it to bring a amino acid to the ribosome, okay, and so it's very good at transferring RNAs or transferring amino acids. Messenger RNA looks like this, and its job is basically to bring a message, and every single one of these little bases right here would be that message. Okay, one thing that hasn't really been shown much, and just to repeat, is the individual carbons that make up these nucleotides. Okay, and so if I draw a individual nucleotide, again, I'm drawing my mouse, so I can't draw very good, but one thing I want to say is this is a carbon, 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 carbon. So if you count those, 1, 2, 3, 4, 5, that is a 5-carbon molecule, 5-carbon sugar. This would be an oxygen. This, like we said, was our phosphate. Okay, so that's a phosphate. This is our nitrogenous base. Okay, and so those bases can be either A, T, C, or G, or U in RNA's case. This is the first carbon. This is the second prime carbon. This is the third prime carbon, fourth prime, and fifth prime carbon. Okay, first, second, third, fourth, fifth. So that's really important because where is the phosphate attached to our sugar? You would say the phosphate is attached to the fifth prime carbon. And where is the nitrogenous base attached to our carbon or our sugar? You would say the first prime carbon. And where is our adjacent or next nucleotide connected to? If we had another nucleotide down here creating a chain of nucleotides, and let's say this is a 2-ring base, which we're going to learn about later. Okay, this would be a purine, and that'd be a pyrimidine. But let's just assume we have this second nucleotide. This would be the fifth, right? Just like above. Fourth, third, second, first, oxygen. Our adjacent nucleotide would produce a bond between this phosphate and that sugar, specifically at the third prime carbon. Okay, so 3 different carbons in our 5-carbon molecule that are really important. The first prime carbon is always attached to the nitrogenous base in every case. Okay, first prime attached to nitrogenous base. Third prime is always attached to the next or adjacent nucleotide in our sequence. The fifth prime carbon is always attached to the phosphate, and so that's going to give us this, like I drew before, this arrow, which means this would be a 5 prime to 3 prime direction, because our 5 prime is our highest prime carbon and our 3 prime is our lowest prime carbon on a particular nucleotide. Okay, and so we're always going in this direction, 5 prime to 3 prime, 5 to 3, then we'd go 5 to 3 on the next one, and we'd go 5 to 3 on the next one. And so what is our overall direction? We are overall going from a 5 prime to 3 prime, and so we don't necessarily need to know how DNA molecules are built, but know that DNA molecules are built 5 prime to 3 prime, and it's because of that. You link a nucleotide to the 3 prime end of each nucleotide, and because you link the nucleotide to each 5 or 3 prime end, you are building the molecule in a 5 prime to 3 prime direction. Okay, and so that's going to be really important, especially later in the year. As long as you know that the monomer is a nucleotide and the polymer is nucleic acid, and you know a little bit about DNA and RNA, specifically the bullets in these questions, or bullets on these slides, and you know the structure of a nucleotide and how nucleotides are linked, then you should be good to go. One thing I'd like to mention too, and I don't know that you're going to need to know it in this chapter, but you will shortly, is that these 2 bonds right here, let me highlight these. This bond and this bond are a special type of covalent bond called a phosphodiester bond. And we'll talk a lot about that in class. You do need to know that bond, phosphodiester. If you look at that name, phospho comes from phosphate, right? They are bonds coming from the phosphate. Phosphodiester, so di means 2. There are 2 of them. They are specifically ester bonds. Okay, so 2 ester bonds coming from each phosphate, you get the term phosphodiester bond. Okay, and like I said, those are special types of covalent bonds. So we know they're thick, or we know they're strong, rather. Strong bonds, they hold the backbone of the DNA and RNA molecule together. But like I said, we'll review this stuff in class. Have a good night. Mr. P, out.